So it's my great pleasure to introduce the next speaker, is Hande Özdinler. You've heard from her already today, so you know who she is. You've seen also in my presentation that she really has done beautiful work on animal models of ALS. One thing I'd like to stress about Dr. Ozdinler, who is an associate professor in neurology here at Northwestern, is that apart from the remarkable experimental work she does on different models of ALS and on the finding of biomarkers and potential cures, she's also a terrific advocate for ALS research and ALS patients. And her tireless work on that has made a huge difference in the entire community for ALS study here in Northwestern. So thank you, Hande.
Thank you so much, Marco, for this kind introduction. So my talk is about ALS pathology in the TDP pathology in the ALS brain. And I think this is one of the most important proteins, as Dr. Kalb has outlined. And this protein is very unique because it lives both in the nucleus and in the cytoplasm. And it plays a role in the nucleus. It also plays a different role in the cytoplasm. So it goes back and forth, back and forth. But there are very few proteins that does that, and TDP is one of them. So what we define as TDP pathology is the accumulation of phosphorylated TDP in the cytoplasm. So when that happens, there are two types of problems. One of them, TDP cannot do what it is supposed to do in the nucleus because now it is trapped in the cytoplasm. So it is the lack of function. And then the second problem is that it is accumulated in the cytoplasm. And by accumulation, it also acts as a sink that it accumulates together with many other proteins. So then it does not allow the cell to do what it is supposed to do. And there is a big protein accumulation problem in the cytoplasm as well. So there are two major problems that goes on inside the neuron. And this problem is present in ALS, ALS FTD, 50% of Alzheimer's disease patients, in Parkinson's. So if we can actually bring a resolution to this TDP-mediated neurodegeneration, our solutions could be more broadly implicated in not just in ALS, but neurodegeneration at large. So that's the reason why many of us focus on the biology and the pathology of this protein. So what happens in the brains which have TDP pathology? So in this study, you're looking at the electron microscopy images taken from upper motor neurons, the neurons that degenerate in ALS patients, and the neurons that actually initiate and control voluntary movement. They are in the brain, they project from the cortex to the spinal cord, and the spinal motor neurons then project to the muscle. That's why the movement starts in the brain, and the brain component is very critical in motor function and also in ALS. So we find that the upper motor neurons in ALS patients who actually have TDP pathology, they have three main problems. In addition to the problems that I told you about, accumulation in the cytoplasm. First, their mitochondria is falling apart. Especially the inner mitochondrial membrane is falling apart. When the inner mitochondrial membrane falls apart, you can't really generate ATP. You can't generate energy. So that's a major, major problem. Second problem is the endoplasmic reticulum, where the proteins are made. They're expanded or they fall apart. That tells you that the protein machinery is also falling apart. And there's ER stress, and then they can't make the proteins that they want. They don't have energy. And then if you look at the nuclear membrane, you can see that the membrane is also swollen. The transport from the nucleus to the cytoplasm has a problem. So imagine that neuron. They don't have energy. They can't make the protein. They have all these accumulations, and the communication between the nucleus and the cytoplasm is also not possible. How can you be happy? And eventually, they degenerate. So now, these electron microscopy images taken from human samples actually tell us the core of the problem. And by generating a mouse model that actually carries the mutation detected in human patients, A315T, this TDP-A315T mouse model was generated, and it very closely recapitulates the human pathology. So when we cross it to our GFP mouse, in which upper motor neurons are genetically labeled by fluorescence GFP expression, we can actually visualize these diseased upper motor neurons. And we can see that they progressively degenerate over time. This allows us to bring cellular clarity to upper motor neurons and study those upper motor neurons with cellular precision. And then we ask them, do you show similar problems as the human upper motor neurons? And they say yes. They say, yes, our mitochondria is falling apart, especially the inner mitochondrial membrane. We have problems with mitochondria, even along the apical dendrites. Our cell bodies are filled with mitochondria that are problematic. Our ER is falling apart. We cannot make protein, and sometimes they are swollen. And yes, our cytoplasm to nucleus transport is defective. So this made us realize that the upper motor neurons in mice and upper motor neurons in men have similar cellular problems in the presence of TDP pathology. And you may remember Dr. Kalb's slide, where is the translation? We think the translation is at the cellular level, that the cells, regardless of different species, they actually face the same problems. So if we can begin to solve their problems, maybe we can make them happy. So that was the idea. That was the idea when we started collaborating with Dr. Silverman, who invented a new compound he called NU9. And this compound was actually identified based on its ability to reduce misfolded SOD1 aggregation. So now, misfolded SOD1 and TDP are two different proteins. But we thought, if this compound can reduce misfolded SOD1 aggregation, and TDP is also another protein aggregation, can it actually work on TDP as well? Because if it is a mechanistic, if it works on a mechanism of reducing protein aggregation, there's reasons to believe that it may work. And after 60 days of administration of NU9, we realized that the mitochondria was actually becoming more intact, and the inner mitochondrial membrane was becoming more intact, and also the endoplasmic reticulum returned back to almost normal. That you could see the cristae, you could see the ribosomes, you don't see the swelling. And then at two different levels, both at the mitochondria and ER, there was remarkable improvement even just by 60 days. So then we said, does this translate to neuronal health? And yes, it did. So after 60 days of treatment, the corticospinal motor neurons that degenerate due to TDP pathology were actually retained in the motor cortex, and we did not observe the ongoing cellular degeneration. And also the apical dendrites that become vacuoled and falling apart, that also stopped. So this was a remarkable finding, because now for the first time a compound was able to help the diseased upper motor neurons regain their health. But many people ask us, how does it do it? Because now this protein works on mitochondria, ER, SOD, TDP, this is like a magic compound. How does it work? Where does it bind to? What is the mechanism? To bring an understanding to this, we actually performed proteomics. And we gave TDP mice NU9 for 60 days, 3 groups, wild-type mice, diseased mice, treated, untreated. And then after 60 days, we took the motor cortex, we did proteomics, and there were about 53 proteins that went from disease state to wild-type state. So it was able to change the expression levels of proteins, 53 proteins. Then we said, okay, proteins. Who are you? Where are you? What do you do? And the ingenuity pathway analysis told us that there was actually related to organismal injury abnormalities, like, these are the structures that I told you about, like mitochondria ER, and then said these must be genes related to skeletal and muscular disorders. And imagine, the computer does not really know what genes I'm giving, right? And then it says there's cellular assembly organization, cellular function maintenance, right on spot. Then we said, okay, show me the canonical pathways, the top ones, what's happening? And it says the fatty acid beta oxidation. And then valine degradation, some amino acid degradations. What does this mean? But I need to tell you that in mitochondria is the organelle that generates ATP. But it does it in 2 different ways. One of them is the electron transport chain, but the other is by degrading the free fatty acids, okay? And that's very important. So it seems as if this beta oxidation, degradation of the fatty acids to generate more energy, and especially when the inner mitochondrial membrane is broken apart, is upregulated. So that could be one mechanism that NU9 tries to fight and tries to change the dynamics by increasing the beta oxidation. And then isoleucine degradation, valine degradation. So what does this mean? These are key amino acids, but sometimes when the cell is deprived of energy, it actually degrades it so that the end product is fed into the methylmalonyl pathway. And then you may say, what is this pathway? It is actually an important pathway for the metabolism of fatty acids. And again, trying to make more fatty acids available for the beta oxidation. So again, this methylmalonyl pathway, the end product is the succinyl-CoA, and you may know, if you study the mitochondrial energy metabolism, that's how you enter, that's one of the amino acids, or that's one ingredients that you need for the Krebs cycle to generate ATP. So it seems with this proteomic study that NU9 treatment enhances energy production via distinct cellular events. And that may be one of the reasons why we see improvement in those neurons. But would improving the integrity of mitochondria and increasing energy production be a solution for reducing and overcoming TDP pathology? So I don't have time, but we have 2 key publications to show you that the mitochondrial dysfunction is a very, very early event in ALS and in TDP pathology. You actually see mitochondrial dysfunction before TDP pathology occurs, before the TDP accumulation occurs. And by regulating this mitochondrial dysfunction, you can actually have improvement in neuronal health, which makes us believe that looking at mitochondria would actually be a good strategy. But then, does this reduce TDP pathology? At the time, we didn't have the tools, but I'm happy to report now that we have 2 different mouse models of TDP. One represents the familial, the TAR4 mice, again, Dr. Kalb mentioned about, and the other one is the TDP-A315T, which recapitulates the familial form. And in both of those cases, we actually have now antibodies to detect the phosphorylated TDP in areas that are involved in neurodegeneration in ALS and ALS FTLD. So we have the tools now to better understand this. And I think the NU9 treatment may be important for TDP pathology at large. And it may be useful not only for ALS, but ALS, FTLD, and Alzheimer's disease. So I'm happy to tell you that we have collaborated with Dr. Silverman and now included Dr. Klein to our team, and we recently received $7.3 million from National Institute of Aging to further study the potential impact of NU9 on Alzheimer's disease and ALS FTLD within the context of TDP pathology. And hopefully I'll be able to tell you more in the coming years. So I want to thank you for your attention. And again, this work would not be possible with the grants and supports that we have received from Les Turner ALS Foundation, Along Swim, NIH, NIA, Brain Research Foundation. I'm thankful to everyone. And I'm also thankful to Society for Neuroscience because they have chosen our GFP neuron and they made a huge big poster. I don't know if you have seen it during SFN. We were thrilled to see it. And I'm also very thankful to all my lab members who are here and presenting posters. And thank you for your attention. I'm ready to take your questions. Thank you.
Can you hear me now?
Yes.
Okay. So there are some questions from the online people. Do you think initiation of antioxidant SOD in inner mitochondria causes TDP-43 mislocalization?
Okay, I can't hear.
Do you think initiation of antioxidant SOD in inner mitochondria cause TDP-43 mislocalization?
Okay, this is a complicated question, but I think the reason that we see the TDP pathology is there are multiple folds to it. TDP also binds to the outer membrane of mitochondria, and when the inner mitochondrial membrane becomes leaky and mitochondrial DNA is secreted or released to the cytoplasm, that creates many events inside the cell that they try to maintain the leakage. And as doing so, they try to accumulate around the mitochondria. So I think the TDP pathology and mitochondrial dysfunction go hand in hand, and I think that's a very early event. In my personal opinion, I think if we can improve the integrity and the health of the mitochondria, we would actually reduce TDP pathology.
Hi, I have a question. Is the NU9 compound can penetrate the brain BBR the brain spinal cord? I mean, you use the oral administration of the drugs. Can it enter into the brain? Can the compound penetrate?
I can't hear it.
Does it pass the blood-brain barrier? NU9?
Oh yes, NU9 plus crosses the blood-brain barrier.
After you administer the NU9, do you have some survival improvement?
Yeah, so we haven't looked at the survival. For those assays, you need, first of all, a lot of mice. And second, with the clinical trials, we have seen that the mouse survival does not really translate to patient survival. And we try to take more cellular approach, that we would like to look at the integrity and health of the neuron. And in our studies, we would like to improve the health of the neuron, and by doing so, improve the circuitry, and then improve the clinical outcome.
Hi, I had a question about NU9 as well. It's not super intuitive to me why something that's designed to dissolve SOD1 aggregates would also work for TDP-43 aggregates, unless it's like a general detergent or something, where there are some structural similarities. I wonder if you could comment on that.
This is a very good question. We also want to understand this. So the same proteomic studies that we have done to the TDP, we have also done in the SOD1 mice. And it seems as if it is a protein aggregation, but the mechanism that leads to that protein aggregation is actually different. But also in the SOD1, you have mitochondrial problems. You also have ER problems. So there are some common themes, and I think we should be focusing our attention to those common themes. And Okay, I have to wrap up. Maybe
No, but
one more
yeah.
question? All right.
Maybe you explained this before, and I'm sorry I didn't understand it, but can you really explain what is the target of NU9 exactly?
Yes, so I think it is not that it does bind to just one thing or does one thing. So I think the target of NU9 is reducing protein aggregation, improving mitochondrial health, and also reducing ER stress. And I don't think it's going to do it by just binding to one protein. And it is possible that it interacts multiple proteins, but down the line, eventually, it actually helps mitochondria, ER, and cytoplasm.
So if you're going into clinical trial, what is the target engagement that you're thinking you're going to be doing?
Yes, that's a very good question. For the target engagement, I think we would be looking at multiple things. And for patients who develop the disease due to mitochondrial dysfunction, due to cytoarchitectural defects, or due to increased protein aggregation. So for each patient, I would imagine that that would be a different biomarker that we should be looking at. This is actually the reason why we received the DoD grant. And we don't think that it's just as simple as, just one molecule or one target does this. It would be possible that we have to look at combinatorial proteins and combinatorial signatures to look for the target engagement.